CRISPR-I squared, simultaneous gene repression by attacking transcription and translation, a novel dual CRISPR approach by the Tulane Synthetic Biology Club. We are the Tulane Synthetic Biology Club, dedicated to teaching other passionate students about genetic engineering through hands-on in-lab experiences. Our current research is focused on silencing gene expression using two catalytically dead CRISPR systems. We chose to use a combination of both dCas12a and dCas13a CRISPR systems. We call this the CRISPR-I squared system. The CRISPR-I squared approach. CRISPR-I squared stands for CRISPR interference, and since we're using two systems, it's squared. Research has been done on the Cas12a and Cas13a systems independently. However, our hypothesis is to test how effective the silencing effects of the CRISPR-I squared system are, and if 100% repression can be reached by combining these two systems.
CRISPR, standing for Clustered Regularly Interspaced Short Palindromic Repeats, is a family of sequences found in bacterial DNA which helps bacteria defend against invading bacteriophages. CRISPR works by using nucleases called Cas, which cut off sections of infecting viral DNA. These DNA fragments are then inserted into the bacteria's own genome. Over its lifetime, the bacteria can take many DNA fragments from many invading viruses, and these fragments of DNA are separated in the bacteria's genome by palindromic repeats. When a virus tries to infect the bacteria again, the bacteria can use these saved DNA fragments and the nuclease activity of a Cas protein to easily identify and destroy it. The DNA fragments act as instructions to lead the Cas protein to a target sequence. Guide RNA, or gRNA, is the actual nucleic acid to lead the Cas9 protein to the target DNA, which will be destroyed. gRNA is made up of CRISPR RNA, or crRNA, which is the complementary sequence to the DNA fragments in the spacer sequences, and transactivating CRISPR RNA, or tracer RNA, which helps structurally support the crRNA. Cas9 will check for a protospacer adjacent motif, or a PAM sequence, which is located downstream the target site on the invading viral DNA. Then it will locate the region targeted by the gRNA and will make a double-stranded break there, preventing the virus from being able to continue its invasion. While Cas9 is the more well-known protein used in CRISPR-Cas complexes for gene editing, we will use dCas12a and dCas13a as they are more suitable for our project.
The Cas12a protein only requires one RNA molecule, the cRNA, because it is able to create this RNA by using its own ribonuclease site. Cas9, on the other hand, requires an enzyme called RNase 3 to create its cRNA. Cas13a also only needs cRNA. Cas12a is also smaller than Cas9 and uses an upstream PAM sequence, lowering the chance that it will bind to the wrong sequence. For these reasons, we use Cas12a and Cas13a. We use deactivated Cas proteins, which are catalytically dead, meaning they can no longer perform endonuclease activity. However, the protein is still able to bind to the target sequence and prevent other transcriptional or translational proteins from binding, preventing the creation of mRNA or proteins. This allows for gene expression to be altered without altering the genome itself. Using this technique, we can explore the effects of a gene without fully knocking it out. Typically, RNA polymerase will bind with a DNA sequence to create mRNA. A ribosome will then bind to mRNA to create a protein. dCas12a systems bind to the promoter region of a gene to prevent mRNA transcripts from being made. If no mRNA is processed, then there will also be no protein product. However, dCas12 is only roughly 90 to 92% effective at this, so some mRNA transcripts will be present and formed. To inhibit the mRNA products that are formed, we employ another system. The dCas13a system prefers to bind to RNA rather than DNA. If a bulky protein is bonded to the mRNA transcript, it cannot bind to a ribosome and begin synthesizing a protein. When looking at the combined CRISPR-I square system, we can clearly see how there are two checkpoints to ensure the gene is silenced. First, a very minute quantity of mRNA product will be transcribed due to the presence of a dCas12a system sitting on the gene. Additionally, most of the very minimal mRNA products that are made will bind with the dCas13a machinery. The dCas13a machinery will then prevent a ribosome from attaching and thus prevent any protein translation. This will ensure that the absolute minimum of protein products are made.
CRISPR-I squared. The objective of our project was to combine dCas12a and dCas13a systems to maximize gene silencing capabilities. In previous experiments, the two systems were isolated and tested to determine a benchmark of silencing percentage. Cas12 represses roughly 92 to 99% of mRNA products, while Cas13 represses roughly 93% of protein products. Testing CRISPR-I squared on GFP and E. coli. A critical aspect of our experiment is the catalytically dead portion of our CRISPR-I squared system. We tested our system on E. coli DNA. The E. coli used had a green fluorescent protein gene, or GFP, encoded within its genome. This makes for an easy target for our CRISPR-I squared system, and to test for efficiency, we can use flow cytometry to measure the percent fluorescence. A lower percent will indicate that more gene suppression occurred.
Step-by-step workflow. Our team developed a library of potential sequences and identified 5 target sequences for Cas12a and Cas13a respectively. The sequences were all part of the GFP gene. We also identified a negative control sequence that does not transcribe for the protein. The Cas12a protein requires a PAM protospacer adjacent motif site in order to bind. However, Cas13 does not. This means that we selected our Cas12a target sites based upon their available PAM sites in our target sequence and selected the 13a target sequences that were closest to the ribosomal binding site. The Cas12a and Cas13a protein combination is catalytically dead, meaning it is unable to cut target sequence like a traditional CRISPR system. The dCas12a system will bind to a specific location on the target gene and stop transcription by blocking any transcriptional machinery from binding. Since 100% repression cannot occur using dCas12a alone, the dCas13a system will then bind to any mRNA products which do not form. This will then stop any translational activity from occurring by preventing the transcript from being read by translational machinery. We pursue the covering approach as opposed to the cutting approach because we believe this system to be a safer, more temporary approach to genetic engineering. Rather than removing a sequence entirely, we can simply prevent a gene of interest from being transcribed until a future event, after which our catalytically dead CRISPR system unbinds from the DNA and the gene is available for transcription. This modularity makes dCas-A systems incredibly versatile and adaptable to various experimental needs.
Assembling the dCas12a plasmid. The dCas12a plasmid that we used was previously verified by a separate study. No other work had to be done on this plasmid. The plasmid used contained chloramphenicol resistance, which is one of the 2 antibacterial resistances used in our experiment. Unlike the dCas12a plasmid, the dCas13a plasmid had to be built. First, 3 regions of catalytic activity were identified within the Cas13 protein. Each of these regions would serve as a potential target to inhibit the activity of the Cas protein. One of the 3 regions was inactivated via a PCR. The other 2 sites of activity were altered using a hi-fi DNA assembly with a G block to contain the mutations. New England Biolab's E5520s was used as a hi-fi DNA assembly. After this, a Gibson assembly was done to ligate the PUC19 backbone and the dCas13a plasmid. One issue that was prevalent is the transformation only picking up one of the 2 plasmids. To account for this, there were some differences between the plasmids, which allowed for selective pressures on the E. coli cell to pick up both of the plasmids. First, each plasmid had a separate origin of replication. This decreases the competition within a cell for these 2 plasmids to be picked up because the 2 will not compete for the same machinery. As a result, each plasmid can be replicated without hindrance from the other. Each plasmid also had a different antibiotic resistance. The 2 differing antibiotic resistances would allow for less competition within the cell and for the E. coli, the NEB5 alpha E. coli cells to pick up one of each plasmid. Typically, during transformations into the NEB5 alpha E. coli cells, one plasmid would be favored over the other. And due to this, the cell would only have a dCas12a or a dCas13a plasmid, not both. To successfully complete the dual transformation, the two separate origin of replications and bacterial resistances causes a selective pressure to be placed on the E. coli cells. This then encourages the cell to pick up both plasmids, and as a result, the CRISPR I² system would be functional, with two plasmids, one dCas12a and one dCas13a, both affecting the genes. Results and future direction. Flow cytometry data on the dCas12a system was collected. The highest repression occurred at G2 and G3. G2 and G3 are two target sites for the dCas12a system within the GFP gene. Data on the Cas13 system is currently being collected. We hope to gather flow cytometry data and find what region represses best within the Cas13 system. After this, we can combine the two systems and determine the repressibility of the CRISPR I² system. Cell fluorescence will be detected via this flow cytometer. If there are low fluorescence levels, this will indicate a high rate of repression, thus leading to a conclusion that the dCas13a system was also successful in silencing GFP. Our team is in the process of collecting further data on the dCas13a's repression abilities, and we hope to use this data to identify which locations are ideal.
Further uses. The potential of CRISPR as a tool for genomic editing is immense. DNA is the foundational code which most organisms rely on to live. Being able to manipulate this code brings about endless possibilities. Agriculture. In agriculture, plants can be modified to become resistant to insects, viruses, or bacteria, become more nutritious, or even increase the shelf life of produce. Additionally, proteins which cause allergic responses can be removed from plants, removing a danger from the lives of many people. Livestock. Similarly, genetic modifications can be made in livestock to protect them from disease, generate a higher yield of product, or create bioactive proteins which can benefit human health. Research. Gene slicing can also be used in research and medicine to help scientists more effectively create new drugs or therapies by allowing them to observe the effects of certain gene knockouts or knockdowns. Medicine could also be used to target cancer cells, which often overexpress certain genes, allowing them to continually divide and escape apoptosis. Ethical considerations. There are certainly ethical considerations to be examined when modifying the genomes of living creatures. We as humans do not fully understand the complex relationship between genes, organisms, and the environment, so we must be careful about what we experiment with. In the Tulane Synthetic Biology Club, we experiment on E. coli, which has been deemed to be ethical, but we still intend to keep the moral implications of our work in mind.